Hello, welcome back to my YouTube channel, My Intuition 4865. This is the class series for biology for engineers other than computer science stream. This is mainly for electronics, mechanical, and civil streams who joined in the academic year 2022 onwards for the Visheshwarya Technological University. Okay, so this class series I'm just started. Okay, BBOK407, that is the subject code. If it is BBOC, then that is for computer science stream. That class series already started, and its playlist link you can find in the description box, as well as the PPT. Whatever PPT I made here, its PDF version also I can find in the description box. Once you can keep watching. Once I upload the video, I will upload the PPT also. It will be helpful for you to refer and study. Okay, so move on to the syllabus which is given here. So this is the module 1 of BBOK 407. K becomes more. Okay, BBOK 407. Okay, so the cell structure, the basic unit of life, structure and function of cell, the plant cell and animal cell, prokaryotic and eukaryotic cells, stem cells and their application, biomolecules, carbohydrate, nucleic acid, proteins, lipids, enzymes, vitamins and hormones. This much is there. So this particular class series I am trying to cover these two this much till prokaryotic and eukaryotic, as well as stem cell and their applications. Okay, so it should be based on the outcome based education. So the course outcomes, as a teacher I must show you. You should read it out. These are the course outcome, what you want to learn, what you will learn after completing this particular class, particular syllabus or particular subject or course. Okay, a question wise question and its answer scheme type video class I take usually now. Okay, so question 1, examine the structural differences between plant and animal cells. Relate these differences to the specific functions such as each type of cell performance. Illustrate with a labeled diagram. A simple schematic diagram is given, that will be more well and good. So this is connected to course outcome 1. Level is 3. Question level is 3 and it can carry 10 marks. Okay, so the answer screen, identification of structure differences carries 4 marks. Relating the differences to specific function carries 4 marks. The diagram, once you draw that, it carries 2 marks. Okay, now the answer, if you see, you can see that in the real life, the uh, the cells which are, they are very small, even very difficult to see with our naked eye. Even it is difficult to see with common microscope, but they are building blocks of the all living things, for the plants, animals, to even the tiny bacteria. So they are like they are the tiny factories which work together to keep the organism alive. That's why they are called the basic unit of life. Okay, now the answer for the particular question given, the plant and animals, their cells, they exhibit distinct structural variances which contribute their specialized functions within living organism. In the cells of plants and the cells in the animals, what is the difference? So we'll see one by one. First one, that is the structural difference. In that first point, the cell wall, the in plant cells, the plant cells are like a tiny house with bricks, uh, which represents the wall. Okay, so this rigid wall, this actually it is made up of a very tough fiber which is known as the cellulose. So this, it gives the cell its shape, and it protect the cell from the very harsh environment, or from the for getting crushed. Okay, so that in case of the plant cells. In case of animal cells, they are like a freestanding stents. They don't have any cell wall, or they can take any various shape depending upon the specific function which comes to that. So that about the first difference with the cell wall, which is identified. Cell wall is there in in case of plant. Cell wall is not there in case of animal cell. And second point, that is chloroplast. In plant cells, these chloroplasts are they are the solar panels. We know what is the function of solar panel, to take the sunlight energy and convert into energy. Okay, same way chloroplast in the plant cell, they are the they contain a chlorophyll, which is a pigment, a color giving substance called as pigment. So the chlorophyll, which is a pigment, it captures the sunlight, and the plants use to make this to create their own food by a process called the photosynthesis. In animal cells, we don't make our own food, we always steal others food. Okay, so we don't want that chloroplast. So normal animals, they don't have the chloroplast. Some microorganisms which comes under the animal kingdom category are having a similar type of one, but in general animal cells are not the so-called animal cell is not having a chloroplast. Then comes the third one, the vacuole. uh, it, in plant we call it as the central vacuole. It's just like a big water balloon in the center of a plant cell. It stores the water, it stores the nutrients, and also the waste products of the plant. So it helps the plant to stay firm, upright, by creating the so-called the turgor pressure, which is like the water pressure inside the cell. In case of animal cells are actually vacuoles are very rare and very small vacuoles are there. They are not nearly as big as or not much importance as that of the vacuolae present in the plant cells. So they may store some material or help break down waste production. Then comes the fourth point, the shape. The plant cells, within cell wall, the plant cells tend to be very uh, say boxy, rectangular. So this shape which helps them to pack together efficiently in the plant tissues. Whereas in case of animal cells, they lack this particular shape. Square shape is not there, even circular shape is also not there. They are random shape, sometimes round, sometimes oval, even irregular, depending upon the job that particular cell has to do. So example, a muscle cell, they are very long and thin for a contraction, while the nerve cells are they are having their own long extensions to send the messages. And okay, that about the shape of both plant cell as well as animal cell. Now we'll see the specific function. The cell wall in plants, it supports, it is made up of cellulose, I already said. So it acts like a skeleton for the plant cell. It's very strong and rigid, and it prevents the cell from collapsing, as well as it gives the plant its the overall structure. uh, Suppose if you imagine a balloon which is filled with water, it may bulge. It may if a balloon, if you fill water, it will bulge. That cell, the plant cell should not bulge, therefore the cell wall make it protect. Okay, it's like a very tight rubber skin, keeping its shape and allowing it to plump. Okay, so that about the specific function of the cell wall. Now the chloroplast explained already that it is in photosynthesis. They are the tiny the power plants within the plant cells, containing the chlorophyll, which is a pigment which captures the sunlight and the water content in the cell through a process called photosynthesis. The chloroplast use sunlight, water, and carbon dioxide to produce energy. Usually it gives sugar, which but the plant need to grow and survive, which we need to grow and survive. So the plants take in the light and air and the they create their own food source. That is about the chloroplast, the duty of a chloroplast. Then comes central vacuole. Central vacuole, it maintains that so-called the trigger pressure. Uh, uh, the large central vacuole, it acts like a multi-purpose storage unit. It's a water reservoir for the plant cell. It holds the nutrients which is needed for the plant to grow. It even also collect the waste product which is produced. The vacuole also plays a very crucial role in maintaining the called trigger pressure. It's like trigger pressure is a water pressure inside the cell. So the vacuole, it fills up the water pushing against the cell wall and keeping the plant cell rigid and plump, which helps the plant to stand upright and grow tall. So without the turgor pressure, the plant stems, it will be very floppy, will not be having the rigidity in it. Then move on to second question about prokaryotic and eukaryotic. Can take it as a side heading, next side heading prokaryotic and eukaryotic, but I made a question for that. Critically analyze the distinction between prokaryotic and eukaryotic cells. Discuss their respective advantages and limitations in the context of biological systems. Again, connecting to course outcome 1, level 3, 10 marks it can be asked. Critical analysis of distinction 6 marks, advantage and limitations of 4 marks. Okay, so we will see one by one. Let us take the images. Okay, no need to study as such the images. A rough sketch if you draw, it will be well and good. Otherwise, just for reference I kept it here. This is not for engineering students as such. It's for biology students. I always repeatedly tell, but to understand things, images are very good. Okay, so an image is given which is eukaryote as well as the prokaryote represented there. So we'll see the point one by one. Cell structure. Before that, the prokaryotic and the eukaryotic, the cells, these two cells, they are two fundamental cell types with their own distinct characteristics, their own advantages and limitations. The prokaryotic cell and eukaryotic cell, they differ significantly in their structural complexity as well as their the organization, as you can see in the images. So the first point, that cellular structure in prokaryotic cells, it lack a true nucleus and membrane bound organelles. Okay, so this is the prokaryotic one, the one with the tail. It's very small size actually. Okay, so they are just like a simple room which contains the genetic material, that is the DNA, all the other substances whatever there, it is just floating in that room freely, just like a jelly like substance in that called as the cytoplasm. So the DNA, the other organelles, everything are there in the cytoplasm, a single row cell which is called as prokaryotic cell. So there is no separate compartment for each organelle. They are all in a single row. Okay, so and they are very small. The total room size also very small. We call it as prokaryotic cell. So the genetic material, it is found in the nucleotide. You can see the genetic material which is present here, reddish brown color, nucleoid. Nucleoid, it is a specific region within the cytoplasm where we can find the DNA. It's a long form. Okay, whereas in case of eukaryotic, it is there inside the nucleus in a round form. Here it is in long form, it is found. Okay, so in case of eukaryotic cell, eukaryotic cell has got a true nucleus which is surrounded by the nuclear membrane. This nucleus, it acts like the control center where the DNA is present within the double membrane. Okay, not even single membrane, double membrane. DNA is very safe. So this separation which protects the genetic material and allows for the controlled access during the cell processes. The organelles, different organelles, including the endoplasmic reticulum or mitochondria, all are present. They are well organized So it just like eukaryotic cell is a well organized factory. The membrane bound organelles they act as specialized departments with their own dedicated functions. For example, the endoplasmic reticulum, which is involved in protein production and the transport. Mitochondria, that generate the energy for the cell. Likewise, they have their own specified place as well as specified role to play. Now, next one, the size. Size prokaryotic cell generally they are very small, around zero point 0.1 to 5 microns. The prokaryotic one. Uh, they are very tiny, which allows them to reproduce very quickly and can squeeze into various environments. That's why we call it as the mainly the viruses. They are prokaryotic. They can squeeze to any environment very fast. They are very small, tiny. Whereas the eukaryotic, they are little larger, of about 10 to 100 micrometers. That means if we are as such, if we treat us as a prokaryotic, eukaryotic cell, if you carry a small marble in our hand, that becomes the eukaryotic cell. So that is the size difference which I want to tell you. So the larger size allows for more complex structures and functions, which becomes but comes with the trade-off of being very slower to reproduce compared to the prokaryotic. Eukaryotic cell division is or reproduction is slow. Then we'll see the genetic material. Genetic material, whenever say that is DNA. In case of prokaryotic cell, the DNA, it is circular. It present in the nucleoid region. The DNA in prokaryotic cell is single loop found within the nucleotide. Okay, whereas in eukaryotic cell, that is inside the nucleus, they are well organized linear DNA. I think previous just I made a mistake, set a mistake. In case of eukaryotic, the DNA, it is linear in structure, whereas here it is circular in structure. So eukaryotic DNA is much longer, and they are organized into multiple linear strands called chromosomes. And these chromosomes are present or protected within the nucleus in case of eukaryotic. Now we need to see the advantages and limitations. Advantages of prokaryotic cell, like the efficiency, the compact structure with minimum complexity allows for the rapid growth and reproduction, which makes them successful colonizers of diverse environments. Wherever they group together, they grow faster and become a colony. Okay, we call it as colonizers. Adaptability: the prokaryotes can withstand very extreme conditions. They can thrive in extreme situations like very hot springs or the very deep ocean. That place also it can survive. Okay, so because they are simplicity, their ability to adapt to their metabolism, because of the single structure, they can adapt as it wishes. Limitations. Limitations of prokaryotic, the complex function, the absence of membrane bound organelles, which limits their ability to perform the complicated cellular functions. They can do only simple functions, and their size constraints, small size, which restrict the amount of genetic material and the specialized structure they can hold, which limit their overall complexity. Now we'll move on to the eukaryotic cell, the bigger one. Advantages like specialization. The organelles, they have specialized tasks, which lead to the increased efficiency and the complexity of the cellular function. and they can perform intricate process like the internal communication, the protein synthesis, compartmentalization. They have wider capabilities, the eukaryotic cells. Limitations, if you see, uh, limitations, the energy consumption, the increased complexity of the eukaryotic cells, they need more energy to maintain all the functions, all the organelles, their cellular process, and the response time, because of the large size and complex structure, sometimes leads to a slower responses to the environmental changes compared to the prokaryotic cell. I say prokaryotic cells can survive in any harsh environment. Certainly they will adapt accordingly, but the eukaryotic cells, the bigger cells, they need more time to change, to adapt to the environmental changes. That about the differences. Now move on to the stem cells. Evaluate the potential applications of stem cells, then in biomedical field, and their role in regenerative medicine. Okay, what is stem cell? The meaning carries 2 marks. Evaluation of the potential application carries 4 marks. The regenerative medicine explanation carries 2 marks. Okay, so we'll move on to the answers part. Okay, the stem cells, they are actually different from other cells in the body. They can divide, or they can form different shapes over a long time, and they are very unspecialized, because they cannot do a special function unless until it is grown to that required shape or size. So they have the potential to become a specialized cell, just like the muscle cell, or a brain cell, or blood cell, whatever cell. These are actually the growing cells which is taken from the living tissues, and it is kept separately in the lab, and whenever needed, it is used to develop a particular cells, a functioning cell which is in need. Okay, these potentials of applications of stem cell in biomedical, so tissue regeneration. Stem cells, they have the remarkable ability to differentiate into various specialized cell types, which make up our tissues. Okay, so the tissues. Let's say if any of the tissue is damaged or injured, or got diseased, then the so-called stem cell therapy, the stem cell therapy, where it introduces these blank slates, the stem cells, which is actually blank, no function, they don't know what to do. But when it is introduced to a place where the other cells are their function, it will copy that and it will grow, and it will fill the damaged area. And if example, if I tell the skin generation after the burning, burn, so the burn cases, the stem cells can be, it can be taken from the healthy skin or other sources, and the stem cells, it can be planted into that place where the burn happened or the wound is there. These will grow there and become a new skin cells, which promotes the faster healing and potentially reducing the scar that burned mark which is there, that will be removed at faster rate. So that is one of the potential application, tissue regeneration. Then orthopedic treatments. Orthopedic treatments, like the bones, the cartilage, the tissue, connective tissues. These are bones, after becoming a little aged, it will having their own wear and tear over time. Even the cartilage also wear tear, connecting tissues, the joints, the joint pain are coming because of the wear and tear, which leading to a condition called the osteoarthritis. Osteoarthritis can be cured with the stem cell therapy. They offer a potential solution to regenerate the tissues and improve the joint function. So example, if I take the cartilage regeneration of the osteoarthritis, the damaged cartilage in the arthritic joints, it can be injected with stem cells. They will differentiate into cartilage cell, helping to repair and restore the joints, giving the required cushioning properties. Then another example application is blood disorders. Blood disorders, the blood disorder, it arises due to malfunctioning or insufficient blood cell production. If it is the, we call it as the blood disorder. So the stem cell, particularly the hematopoietic stem cells, they found in the bone marrow. It can be a source for the healthy blood cell regeneration generation. Okay, so the hematopoietic stem cell transplantation for the condition called as the leukemia treatment, hematopoietic stem cell are used. For the bone marrow, it is uh, uh, become unable to produce healthy blood cells. Then stem cells are injected there. It becomes a life-saving treatment. From a matching donor, we have to take the stem cell, and it has to be infused into the patient and allow them to repopulate their bone marrow and resume the healthy blood cell production. Then about stem cells in regenerative medicine. Regenerative medicine, mainly the organ repair, like the organs like the heart or liver or kidney, they can be very significantly affected by the injuries or diseases. So the stem cell therapy, it is mainly aimed to help or to regenerate the potential stem cells to repair and restore the functions to the vital organs. Okay, the heart cells, the cells present in the heart, similar type of cells only need to be grown there, then only the required function will work. Same similarly for liver as well as kidneys. Then transplanting completely, these stem cells can be used. Okay, so say example, the heart tissue generation after the heart attack, the myocardial infraction. A heart attack occurs when the blood flow to the part of the heart is blocked, which causes the tissue to die, or the tissue death. So researchers are studying the use of the cardiac stem cells, which are found in the, yes, small members within the heart, to regenerate the damaged heart muscles. So these stem cells, it could be a very potential to rebuild the healthy tissue and improve the heart function after a heart attack. One more point, that is neurological disorders. Neurological disorders are caused by the malfunction or dysfunction of degeneration of nerve cells in the brain and the spinal cord. So stem cells offer a very potential role or avenue for the treatment for these conditions, to promote the damaged or dead nerve cells, or to repair it or to regenerate it. See the neural stem cells for the neurodegenerative diseases, mainly the Parkinson's disease or the Alzheimer's disease. They are characterized by loss of specific type of neurons only. Okay, so the neuron stem cells can be used, uh, which is actually there in the brain. It can be used to replace the lost cells. So by transplanting or by stimulating the growth of neural stem cells, scientists, they hope to promote the regeneration of new neurons, which is potentially slow down or even reverse the progression of these neurodegenerative diseases. Oh, that's about the first class of BBOK407. I think it's okay for you. 407, 407. Okay, so please be my valued subscriber. Your support is needed. Please do share this video to others, your faculties or students, or whoever want to know about the biology for engineers interested to them. Please do share my videos. I need more views from your side. Thank you. My YouTube channel name, my intuition 4865. Myself, Dr. Prasad.